Hello and welcome to GeneLab Chats. Today, we'll be interviewing Dr. Pedro Madrigal from the European Bioinformatics Institute located in Cambridge, United Kingdom. We know that spaceflight conditions have been shown to change antibiotic resistance in microbes and sometimes their pathogenicity. Because the health and treatments available to those on board the International Space Station is an area of essential concern, it's important to accurately characterize antimicrobial resistance. In his publication, Dr. Madrigal and his colleagues analyzed several GeneLab datasets to assess methods by which antibiotic resistance is gauged. In this interview, Dr. Madrigal will discuss this research and highlight how GeneLab's data systems and the AWGs helped enable this work. Welcome, Dr. Madrigal.
Thank you. A pleasure to be here.
Let me start off by asking you, could you please let the audience know what were the most important findings of your study?
Yeah, so to give you a bit of context, this project started because we were interested in antimicrobial resistance, which is going to be a huge health problem on Earth. But the microgravity, confinement, and radiation conditions in space can alter it. For instance, the higher levels of radiation could trigger mutations in bacteria. Also, monitoring the risk is of special relevance to space agencies because the access to antibiotics and medical assistance will be limited during deep space travel. To investigate antimicrobial resistance in space, we used metagenomics data from the Microbial Tracking Project. This data set contains DNA sequences of hundreds of bacterial isolates obtained from different surfaces of the ISS, which is the International Space Station. Then we applied deep learning, which is a machine learning technique, to identify the presence of antibiotic-resistant genes associated to different antibiotics. Thanks to that, we were able to identify strains from 2 Enterobacter and Bacillus species that look the most pathogenic among all the species. Then scientists at JPL performed the lab experiments, specifically antibacterial susceptibility test, on the isolates of the species that look most pathogenic and confirmed the computational predictions.
Thanks for that important work that you've shared with us. My second question is, how did the GeneLab database and tools help assist you in your work so that you could achieve your goals?
Yeah, so GeneLab was crucial for this research. First, of course, its database stores all the datasets that were used in this research, and all the data is public and easily accessible. Second, the multiomics analysis working group of GeneLab acted as a catalyst to discuss the ideas and bring the project forward. And it is only by the combination of these two factors that allow the success of this project.
Thank you, Dr. Madrigal. This was a very informative interview. For our viewers out there, you can learn more about this work, how to join the analysis working groups, and access all the data sets analyzed in this work with the links provided in the description section below. Thank you all and have a wonderful day.